In the previous class, we talked about the we were interested in understanding the overall anatomy of the digestive system. We talked a lot about the smooth muscles, which form the bulk of the system, and their role in propelling the food. We did a bit on the peristalsis movement, and then we also talked about what? About the regulation of the peristalsis by way of the enteric nervous system, the two components, and the control of the enteric nervous system by way of the autonomous nervous system, sympathetic and parasympathetic. So, we did a bit of that, and we will move on. Actually, when we talk about the entire system, we can safely divide into three components. The one that we have already done, just the anatomy of it and how the food is propelled along the length. And the second thing is we can talk about the second point we can talk about is the secretions. You see, there are continuously the food has to be digested. It has to be mixed with enzymes, the proper enzymes in proper quantity, and the enzymes keep on changing, the enzymes keep on changing as the food keeps on changing. And then the most important part is depending, you need to provide a proper environment for an enzyme to act, which means the pH. Okay, you have to keep on changing the pH. Okay, and this would be the second component, and the third and the last component would be absorption. Okay. So, right now another this or maybe next lecture we will talk upon how the secretions, okay, what are the secretions, why are they secreted, when are they secreted, and how are they regulated. Okay, see, so how does a particular enzyme come when it comes, and how does a particular acid or alkali is discharged into the system as and when required. So, one thing that is for sure is one of the most common secretions all animals across, okay, is of course you want to keep everything wet, okay, everything wet. I mean, just as we need to keep our trachea, okay, and the entire passage moist, okay. So, we need to actually so how do we manage that? We keep it by way of what you call as the mucus, okay. Mucus, so there is right from your mouth, so the entire length of the alimentary canal throughout you will have a very large number of what you call as the goblet cells. What do you call them as? Goblet cells, okay, and those goblet cells, they are mucus secreting, and mucus it's watery fluid which is very rich in polysaccharides, and little amount of proteins are also there. I mean, not that the mucus if from beginning to the end is same, no, even the quality of the mucus keeps on changing, it's slightly different, but it does many important functions, like most important, it does not allow the food to directly come in contact with the inner wall of the alimentary canal, no, that is a bad idea. Okay. So, it protects it mechanically protects the stomach, and then it's a great lubricant, okay, so that the food can just pass along. It allows the formation of chyme, very important, it is a buffer, okay, and it has simply go ahead, I will move it. So, let us begin at the level of the jaw, okay. You see, you can't really chew the food without saliva. Saliva is very important. Why saliva is important? Because it's only when we chew the food and it's broken into smaller fragments, and then it's dissolved in the saliva, then only your taste buds can have access to the food. So, your taste buds by which you can taste, okay. Taste, hello, salt, sweet, what else, bitter, what else, sour, very good. So, spicy is not a taste, it is not a taste, okay. Bitter. Have you heard the word umami? What is it? Umami, okay, we will talk about this sometime later. So, we are talking about parotid glands, sublingual glands, submandibular glands. So, they are in the upper jaw, they are in the lower jaw, and they secrete a lot of substances, and they contain one enzyme called as alpha amylase, and then it has some lysozymes which will protect us from the bacteria that we might ingest. And you know something, the salt concentration of the saliva, sodium chloride, is relatively very low, Can you guess why? Answer is very elegant. The answer is, if it were as salty, it means the saliva were as salty as blood. Blood is very salty. Do you agree with me? blood is very salty. If that were so, then our taste buds for salt would get habituated. If you expose a particular receptor to the same stimulus continuously for a very long time, the receptors gets habituated, point number 1. And if the salt receptors for salt on our tongue were to get habituated, we will not get the taste of salt when we are eating it. So as to make sure that your sensory system for salt remains sensitive to salt, you make sure that the saliva is poor in salt, so that when you take salt, you immediately realize, no, the salt is too little or too much or whatever. And salt is so important, okay, so important, sodium chloride, I don't have to, it's so important, okay, that nature has made sure that we are very particular, our sensory systems are very particular about the ingestion of salt that we do. Get the point? So, what the point author is trying to make is that the sodium chloride in the, it's not only that, even the osmolality. Okay, so what is it? Osmolality of fluid 300, am I okay there? Hello? 300, okay. Extracellular fluid 300, blood 300, lymph 300, intracellular 300, as the saliva is being secreted in the salivary gland 300. Okay. But as it is being processed by the duct, I will show you how it happens here. As it is being processed by the duct, the lot of ions are reabsorbed. Okay, so here we have the salivary gland. Can you appreciate those glands there? Each component there, each unit there is called as an acina or acinus. The same word we will also use for the secretory component of the pancreas. We will talk about it later. So can you see that? So this is a cup, this is a cup, that is a cup, and this the cup is made up of single layer of cells. And the moment there is a single layer of cell, there is a apical side, hello, and there is a basal side, okay, are you with me? And on the basal side you will have a capillary somewhere there, and these are ducted glands. So the secretions are poured into this duct, there are hundreds of thousands of them, and then the secretion will finally be brought into the mouth, and then okay. Now the saliva as it is being formed, okay, is called as, okay. So these cells will secrete saliva. How they secrete, we will see in 5 minutes. What do? They secrete this juice, okay, which contains certain amount of bicarbonate ions, sodium chloride, enzymes, amylase, etc. And as it goes, so as it is being secreted right in this pocket, osmolality is still 300. How much it is? 300. We will call it as primary saliva. What do we call it as? Primary saliva. By the time the primary saliva flows into the ducts, ductules and the ducts, okay, a lot of sodium, okay, and chloride is absorbed back, and osmolality is almost reduced to 100. Reduced to what? 100. And then we call it as secondary saliva. What do you call it as? We call it as secondary saliva. Now, this is interesting. There is a basal rate at which the saliva is being secreted by our salivary glands, and that keeps on flowing in our mouth, keeps on flowing. The rate may go slightly up or down, but it keeps on flowing, and that is of course the reason why our mouth is always wet. Okay, it may go up when we start eating or when we smell food, particularly when I am hungry, all that would happen, but there is a basal rate. Okay. Now that basal rate happens to be very important, because if the basal rate goes down, it is still there, saliva is there. If the basal rate goes down, then you start experiencing dryness of your mouth and throat. Okay. And then that dryness is conveyed to the brain, and that expresses as thirst. Are you getting the entire story now? It is beautiful. I mean, it does not mean that saliva has stopped being secreted. No, that is very bad. That saliva has to, even the rate at which the saliva is being secreted, if that goes down, okay, dryness of the mouth means generation of thirst, which means behavior, you will start looking for water. Okay. Now, let us look at, I hope this image is clear to you. We are okay. So, what am I doing here? I am okay. This is a single acinus. Can you see that dark brown patch there on the other side? That is a smooth muscle cell, a bundle of smooth muscle cells. Can you see a smooth muscle cell? Smooth muscle cells. What would be the significance of these cells?
You are taking the food and you want modulating more in your mouth.
So you are right. So they will contract and push, eject. Okay, suddenly to make sure that if you suddenly require, you see they will. So okay. Now let us look at let us go for the most important part. So what I will do is I will pick up a cell from here. and I'll blow up the cell here, and the cell has a luminal side. Therefore, as usual, we have done this 100 times. This is the apical side and this is the basal side, and the moment I said basal side, you should immediately look for sodium potassium pump. Done. Okay, we are very familiar with that. Okay, sodium potassium pump, and then every time there are 3 ions of sodium that go in, 2 ions of potassium that come in, and this actually potassium ion keeps on circulating, because there is a channel for there is a channel for potassium. It keeps on secreting. Okay, what is the purpose of this? The purpose of this is to make sure that there is excess of sodium there. As a result of that, you need to keep on make sure that this import system, what's the import system? Look at it and tell me what is the name of the system. It is there. NKCC. What it is? Oh, so familiar. Look at the beauty of the nature. Same protein system from here. It's same. NK system keeps on working, and that keeps on driving. One sodium, one potassium, and chloride. Okay. Now, so we have excess of chloride, and then that excess of chloride is delivered on the apical side or on the luminal side by way of a channel. By way of a chloride channel. By way of what? A chloride channel. Now, a quick word on chloride channels. Okay, they are all proteins. If you look at the entire it's a big family of proteins, chloride channels. You can broadly divide that family into 1 and 2 groups. Okay, one of them is CFTR. Hello, CFTR. Don't give that strange look. I talked about the CFTR in this class, or I haven't. Cystic fibrosis transmembrane conductance regulator. Okay. Sorry. I keep on mixing the classes. I'll explain it to you. There are 2 types. Okay, there are 2 types of channels. One of them are called as transmembrane. And either you are lying or I am lying. One of the 2 is very smart. He is indicated. Okay. Good. Anyway, so one family is called as you get that type of channel, and that type of channel is abundantly present in your respiratory channel called as CFTR, and cystic fibrosis, and the other one is and this is of the other type. And basic question: what would be the difference between this chloride and this chloride transport system and this chloride transport system? The answer to that: this chloride system works protein system. That work goes in a cycle. With one cycle, one protein molecule will take in three, one sodium, one potassium, chloride and chloride, take it from outside and put it inside. Again, it will go through a cycle. But this guy here is a window. Is it always open? No, it's not always open. It's not always open. It is only when you are either ingesting food or thinking of food, and when the salivary glands are excited, okay, and then it starts secreting, then this chloride channel will open like a window, and it will allow I don't know, maybe 50,000 ions of chloride to pass through every second. Huge amount. Okay, so this is a channel. It is a voltage gated channel. Which means what? When this cell is excited. Why excited? Well, it is excited because autonomous nervous system. It is excited because the parasympathetic nervous system input comes. So, parasympathetic nervous system input comes, it is excited. Once it is excited, there is change in the voltage of the plasma membrane. As a result of that, this voltage gated chloride ion channel opens, and it allows the flood of chloride ions from the inside to go towards the luminal side. And why is it? It is because of this. So, have you understood the steps by which a huge amount of chloride is suddenly being released only when the cell is excited, and the cell will be excited when the food will arrive in the stomach. I am sorry, not in the stomach. Okay, good. Okay.
Sir,
yes.
so the saliva have 100 milliosmol of
Osmolality.
and your cells have 300. So won't this
The
affect the water movement?
you are forgetting important step. That is the primary, that is happening at the level of primary saliva, where the osmolality is 300. So when that phenomena is happening in the previous slide that we saw, it's everything is at 300 milliosmoles, that is primary saliva. Then the answer is here. Okay, so these are the primary secretory cells, and these are the cells of the duct. Okay, so here it is 300, here it is 300, here it is 300. By the time the saliva travels down the duct, okay, it is processed. And in the process, what happens? See, to make this to simplify the things, the author has taken one cell here and he has blown up the cell here. And again, in this cell on the basal side, you will immediately get the pump. And on this side, you have you are absorbing lot of sodium. Okay, and the logic is here. You are reabsorbing sodium and this thick arrow, and you are also releasing some potassium. But look at the thickness of the arrow. The author is trying to codify what? This is much more prominent than this, and as a result, as the saliva goes, the osmolality is reduced to 300. But that happens much later at the level of the duct. Does that answer your question?
So it is becoming back to 300.
No. This is 300. By 200, 100, 100, 100, and is released in the mouth 100. It has that's the point I already made, that sodium has to be less. Sodium has to be less. Water has to be there, but sodium has to be less. And because water has to be there, because water is not there, then it will create the dryness. So the volume of the water has to be same. Osmolality has to be less. That is because of less sodium. Okay, and then that's your final formation of saliva. Yes.
Cells of the mouth wall also have like osmolality of 100 milliosmoles or 300.
Okay. Oh yeah. Oh good. So now I understand your question, and I'll give you the answer. The answer to that question is the cells that are lining the mouth, within them they have 300 milliosmoles, but the saliva is 300 milliosmoles. That's absolutely correct. That's absolutely correct, and there are no aquaporins anywhere. Okay, no, good you asked. Otherwise, I had forgotten to mention this point. There are no aquaporins, so water doesn't go across the cells. Water does not go. Good. Okay, so now let's see. This is I'm sure you can imagine. You can immediately see the human face there. From the human face there, can you see the nose there? Okay, so in the nose, so this is what are these? These are the olfactory receptors in your nose. These are the olfactory receptors on your tongue. And the author has given the code for sensory fibers, which is what? Orange color. So the nervous information that is coming either from your nose or from your tongue is going to the olfactory system. Hello, are you good there? And then it is going there. It is going to the hypothalamus. So hypothalamus knows how, by way of your smell and by way of your taste. And then the hypothalamus will send the information to certain groups of nuclei, which are again sitting in the medulla oblongata, and those centers I will call as, what are they? They are neurons. What are they? They are neurons. Okay, they are neurons. All right. What is the peculiarity of these neurons? They are getting information from the hypothalamus. What kind of information? The hypothalamus has already processed the information with reference to taste and smell. Taste and smell. Okay. So the hypothalamus is getting information from where? From the tongue. Okay, which is fifth cranial nerve. It is getting information over the nose, first cranial nerve. Hypothalamus is integrating that information. Okay. It also goes to the memory centers. So you already know that this smell means this particular food. Are you with me? Okay. And then that information will go to the salivary centers. These are the neurons, and these neurons are, as you can see, look at the green line, they are innervating the salivary glands. Is the picture very clear to you? I am trying to make it as simplistic as possible. Okay, yes or no? We are good so far? Good. And then this information is going how? This is sympathetic and parasympathetic activation. Now, see, when we talked about heart and sympathetic and parasympathetic, okay, we gave an impression that the heart is there and the sympathetic nervous system is push it up, make it fast, make it strong, and the parasympathetic nervous system, okay, if it goes too much, and then maintain it within a reasonable limit. Okay. That is true for heart, but there are many places where the sympathetic and parasympathetic nervous system work hand in hand. You get my point? Not in opposite direction, but in hand together in the same direction. And I put this slide because this is a classic example of that. And how it operates, let's see very briefly. Again we are looking at the same cell. What cell is this? This is the cell of the salivary gland. Are you with me so far? But this is interesting because on the basal side, the author has clearly shown two different types of receptors. Okay, and what kind of receptor is this? M1. What does M stand for? Muscarinic type 1. There are M1, M2, M3, M4, M5. Let's stop there. M5. Out of that M1, and they are all G protein coupled. So they will the receptor will go. How many times it will go up and down? 7 times. How many times? 7 times it will go. This and this is coming from where? What is this fiber? This fiber is postganglionic parasympathetic, and therefore it contains acetylcholine. Are you okay with my flow of thought? Okay, preganglion is also acetylcholine, postganglion is also acetylcholine, and that acetylcholine neurotransmitter which is coming via here. These are, I mean, this is something like this. You are sitting in the medulla oblongata. In the medulla oblongata, you have a group of neurons. I will call it as salivatory center. Okay, that neuron will talk to the cells. Okay, and those cells will give rise to axons, which will go over the vagus. Hello, go over the vagus. Yes or no? Yes. Go over the vagus. From the vagus, they will go to they go very close to the salivary glands, but they will have a ganglia. And the postganglionic are also cholinergic, and this is releasing acetylcholine. Now, can somebody tell me what is the meaning of this part? On the smooth muscles that we have seen in the previous slide, smooth muscles, and the smooth muscle cells on the plasma membrane have another protein. That protein is M3. What does M3 mean? Muscarinic. What? But it is not. It's not M1. It is M3. Okay. And then when it is stimulated, those smooth muscle cells will contract and they will expel okay. So the author has given contraction, and then there is also norepinephrine. Now this is a part of sympathetic nervous system. It will act by what kind of receptor? Beta 2 adrenergic receptors, and whereas the M1 receptor, it acts on endoplasmic reticulum. That endoplasmic reticulum will have calcium. When it acts via IP3 mechanism, it will release calcium. Okay. Now that calcium, this calcium is responsible for driving these vesicles, and what are they? They are secretory vesicles. What do they contain? You remember amylase? Alpha amylase is an enzyme. Okay, and then the liquid part, and what all are the components of the saliva, they are being released. So beta adrenergic mechanism, G protein coupled mechanism, it will act via cyclic AMP mechanism, adenyl cyclic AMP mechanism. It will bring about and it will drive the proteins and the mucin. Okay, which is the component of the saliva will be released into the cup, and then it will be driven to the mouth. Whereas now we see whereas this system. Which system? Parasympathetic. What is this? Whereas the parasympathetic nervous system plays this component, see, you need trafficking within the cell. Yes or no? You need trafficking. For that trafficking will not happen without. Good. Whereas this part is stimulated by acetylcholine. This part, beta adrenergic part, sees to it that there is more mucin in it. The mucin component, which is a very important component of the saliva, is more because of the stimulation of the sympathetic nervous system. So are the two systems working against one another? No. Okay, actually parasympathetic nervous system is making sure that the calcium is there so that the trafficking and release can happen. Acting via M1, acting via M3, it is acting on the smooth muscles to make sure that it is ejected, and the sympathetic nervous system is acting on the secretory cells so that the composition of the saliva is regulated. So they are working hand in hand to make sure that saliva in its right concentration with right ingredients is being released at the right time. You have a question? You can ask me. Yes. Is active
since both are working together?
I don't really know. what I know is that as soon as these as you see the regulation is at the level of the salivary center, and once let us see once you either stop eating, or why do you stop eating? Because there is a satiety mechanism. Satiety mechanisms means your system tells the brain that okay, I have had enough food and it is not a good idea to continue eating, so I have to stop eating. Okay. At that time, in that loop, the salivatory centers are also inhibited. Okay. I do not know whether there is any GABA signaling happening there, but at that stage there, and once that is inhibited, the whole system is inhibited, means it stops functioning. Okay. So, you have taken food, you have chewed it, you have mixed it with saliva after as a result of mastication, and the food will go. If you are particularly standing, it is very easy, the gravity will pull the food down the esophagus, and then so this is the esophagus and food will go, and at the junction of the esophagus and the stomach you have a sphincter muscle. We have already seen the control. What mechanism was that? What mechanism was? Latch mechanism. What mechanism was that? Hello? That word is familiar? Latch mechanism. Okay. So this will open and it will allow the passage of the food. So, what we are looking at is inside the stomach, the inner wall of the stomach is thrown into what you call as ragi. What is the word for this? It is called as ragi, and then so this is the esophageal sphincter, this is the pyloric sphincter. After the pyloric sphincter, the stomach will communicate with duodenum, and let us now listen. This is going to become very interesting. When I say very interesting, it is my gentle word for saying that it is going to be complicated. Okay. So please be with me, do not miss a single word. Okay. I will try to make as simple as possible. Okay. Okay. So let us see, we will call this part of the stomach, okay, as the fundus. This part, so the food arrives where? In the fundus part, and the latter part, which is towards the duodenum, I will call as the antrum. Have you observed these two words? What are we are just dividing the two. Traditionally, this is the fundus part, the bigger part, and the latter part is called as the antrum. Okay. Remember this. Now, with this, let us take a step forward, and okay. So now, this is a section through the wall of the stomach, and it is familiar. The outermost layer is serosa, inner layer is longitudinal layer, in the inner layer is a circular muscle. Stomach is more complicated, there is one more layer which you do not get in other parts, it is called the oblique muscle layer. I mean, it is very necessary to make sure that the stomach wall goes through rigorous contraction and relaxation, so that the food is thoroughly mixed with the acid and the enzymes. Therefore, the wall is really thick, and then I will tell you what. If you are walking on the inner wall of the stomach, okay. So there is a hole there, and a hole there, and a hole there, and a hole there. Okay, are you with me? And these are called as gastric pits. What do you call them as? Gastric pits. So can you see that gastric pit there on the surface? Okay, they are very tiny. Okay, very narrow and very deep. They are very narrow and very deep. So they go as far as here. Are you with me? What do you call them as? Gastric pits. Some people also like to call the whole thing as a gastric gland. Okay, but the gastric gland is